So welcome everyone. Welcome to our seminar from the Cancer Cachexia Society. We'll be waiting for a few seconds to have everyone entering. We have more than 120 registrants for today, so it will take a few seconds to let everybody in, and then I'll start with the introduction. So welcome to our Cancer Cachexia Society webinar. Just waiting a few more seconds to let everyone coming in. The number are still growing, so I'll wait for a few seconds more. Welcome everyone, welcome to our seminar. We'll start. So welcome everyone, we are very happy to see you back for a new seminar. So my name is Laure Bindels, I'm an associate professor of pharmacy at the UC Louvain. And so today I'm co-chairing with Dr. Pauline Morini. And so Dr. Morini is joining us, has joined the Education Committee over the last few months.
Good afternoon. Yes, I'm Pauline Morini, and I'm a senior postdoctoral researcher at the Institute for Metabolic and Cardiovascular Diseases at INSERM in Toulouse, France.
And so today, both of us will be co-chairing this presentation. And so before giving the floor to Dr. Rohm for a presentation, I would like to share a few announcements from the Cancer Cachexia Education Committee. You confirm me that you see my screen? Perfect.
Yes.
So the first event that I would like to promote is that to remind you that we have a mentoring program for our trainees member. And so if you want as a trainee to enhance your visibility in the field of cancer cachexia, then you are welcome to send your information and bio sketch to our admin email, and we'll be happy to feature your CV and your picture on our website so that you can get known in the field. And especially if you're looking also for a job, we have job at section on our website. So take advantage of that. Then we are very happy to let you know that our next seminar series will be a trainee talk that will be co-chaired by Dr. Morini and Dr. Pin. It will be on April 8th at 12 AM EST and 6 PM CET. And we already have two confirmed speakers. We will listen to Dr. Chiara Ninfali from Spain. She will be speaking about a 3D model that combines a skeletal muscle component coming from the patient with 3D pancreatic cancer organoids. And then we will also listen to Dr. Noah Forrest, which is doing his PhD at Northwestern University in the United States, and is completing its PhD right now that's been focusing on machine learning model to retrieve electronic records information, and that could help to phenotype cachexia stage. And so we'll be listening to these two very attractive talk, and we have a third one that should be confirmed soon. And we also have a next session of trainee talk in September. Then in June, will have Dr. Julien Gondin that will be coming to speak about his research on neuromuscular electrical stimulation and how it can be used to limit muscle weakness and wasting, and how it can modulate satellite cell function and niche in cancer cachexia. So stay tuned with the LinkedIn and your newsletter or the email from the Cancer Cachexia Society. You'll get the opportunity to register. And then without further delay, it's really my pleasure to introduce Dr. Rohm. So thank you very much, Dr. Rohm, to have accepted our invitation. So Dr. Rohm studied biology in Heidelberg, Germany, and then in Manchester, UK. She then went back to Germany to do a PhD, and she studied adipose tissue lipolysis in obesity and cancer cachexia. And she has been awarded a Novo Nordisk postdoctoral research fellowship that allowed her to join the University of Oxford, where she worked on metabolic change in the context of pancreas, if I remember correctly, using multi-omics approach. And then in 2017, Dr. Rohm joined the Institute for Diabetes and Cancer at the Helmholtz Center Munich, and she's been based there. She's now head of the research division, tissue cross-stroke. And so her work on understanding tissue communication and metabolic reprogramming in cachexia has won her several prestigious grants, including an ERC starting grant and also several prizes. She's very much engaged in the field, in widening the research and knowledge about cachexia, and especially as a board member, board director of the Cancer Cachexia Society. And I would say her axis of research that she will for sure explain much better than me today is exploring cancer cachexia as a systemic metabolic disease and to understand how this tissue crosstalk can be used as a target to enable targeting metabolism and to improve cancer treatment. So without any further delay, Dr. Rohm, floor is yours. We are listening to your presentation. Thank you very much for joining us today.
Thank you very much for the really kind introduction, Laure, and Pauline as well. It's really a great pleasure to be here today and to present some of our work. Let me quickly share this presentation. Okay, so you should now see it in the full slide mode. Yes, great, perfect. So let me hide everything that needs to be hidden. Good. Now, my talk today will be on multi-tissue coordination and communication in cancer cachexia, and I'll be mostly speaking about two very recent projects that we had in the lab. My lab is based at Helmholtz, Munich, so this is a research center focused on metabolic and environmental health, mainly on diabetes. And I think, you know, this already tells you kind of the angle from which I'm approaching cachexia as really a metabolic disease, a problem of metabolic dysfunction. You know, really, cancer can be seen as a multi-organ metabolic disorder. And I'm saying this because it's known by now that tumors do not appear in isolation, but they act on multiple different tissues, including the brain, to cause anorexia, on cardiac and skeletal muscle to cause catabolism and functional decline, to act on adipose tissues to cause impaired storage, and they also activate inflammatory systems, activating chronic inflammation. This is driven by tumor and host-derived mediators, and over the past couple of years, we've been quite active in that field. And the summary of all this, the net effect of these tumor-mediated systemic problems are an imbalance in energy homeostasis, causing severe weight loss, wasting, and weakening of the body, or cachexia, as you all know. And this is really an inexorable, incurable, and deadly disease, as can be seen by these pictures from Steve Jobs, who passed away from pancreatic cancer. And these pictures were taken very few years apart. And you can see how much body weight was lost here, and how detrimental this situation was in the end. So it is a huge medical problem, but of course in this community here, I'm preaching to the converted, so I won't say much more about that. Now, the official consensus definition of cachexia is that it's an ongoing loss of skeletal muscle mass that cannot be fully reversed by nutritional support and leads to progressive functional impairment. This official consensus definition was phrased 15 years ago, and ever since then, the research field has really been growing a lot. Cachexia is very problematic as it affects anti-cancer therapies, and there isn't any current therapy or treatment that prevents or reverses cachexia. And in numbers, we have up to 80% of cancer patients that are affected by cachexia. Prevalence is higher in some cancers than in others, particularly high in pancreatic cancer. And then it's considered to be the cause of death in 30% of these patients. So by these numbers, you can see that it's a big unsolved medical need in oncology. And we see it as a disease within a disease. So it's really something that needs to be treated in order to enable the treatment of the underlying disease, which in the case of cancer cachexia, obviously is the cancer, the tumor. And up here, I've highlighted that cachexia cannot be reversed by nutritional support, because this means that it's not simply a problem of not eating enough or a problem of anorexia, but it's really a problem of metabolic reprogramming. So it's a problem that something within our body, some kind of systemic metabolic programs are affected, are altered by cachexia that cause this wasting and that potentially also amplify this wasting. So very recently, together with my colleague at Helmholtz Munich, Mauricio Berial-Diaz, we have written this review in which we described cachexia as a multi-level disease, as a multi-level mechanism. And what do we mean by this? Well, of course, up here and on the highest level, cachexia is caused by the tumor. That's the primary disease, and that's the initiator of cancer cachexia. But then there's more to it, and we cannot stop by simply targeting tumor-secreted factors. There are, as a second level, host responses. So these are responses by the patient's body that independently contribute to peripheral wasting and further amplify active catabolic processes. And one example here is inflammation. So if we have this low-grade inflammation activated by the presence of a tumor, this will consume a lot of energy. And this will ultimately, of course, contribute to the negative energy balance typical for cachexia. And then at the lowest level, we have tissue crosstalk. So how tissues communicate with each other, how organ-secreted proteins or metabolites might signal to other organs or tissues, and thereby, once again, amplify the wasting signal. And these lower two parts of this multi-level mechanism are what interests me most. And the key points of interest in my lab are metabolic reprogramming, coordinated tissue responses, and tissue crosstalk factors contributing to cachexia. So if we look at CT scans from patients with cancer, we can see that in cachexia, we frequently have muscle atrophy that can be seen here by this red-colored skeletal muscle area, which is much lower in this picture on the right comparison to a healthy person on the left. But in addition to that, typically we have adipose tissue loss, and adipose tissue is shown here in blue and in violet. And that is and a very low BMI. So that is typical that in cachectic patients, despite that not being part of the consensus definition, there is also adipose tissue loss. And we like to study cachexia using typically a combination of many different models, but our favorite model is the C26 mouse model of cachexia, and many of you will know that. But still, I will run you through the most important things here. So cachexia is induced by C26 cells. These are mouse colorectal cancer cells, and they are implanted by subcutaneous injection at the flank of the animals, and then these tumors grow. And within 2 to 3 weeks, they induce body weight loss, as shown here in dark red. And that's typically, in our case in Germany, we have to end the experiments at around 10 to maximum 15% body weight loss. And then we have several control groups. Typically, we have a pre-cachectic group. So this is in rosé here, where we implant C26 cells, but the animals are prepped before they lose any weight. And then we have the non-cachectic group in blue, where we implant C26 cells that never induce cachexia. And we call those NC26 cells. So they never induce cachexia. It's a non-cachectic tumor-bearing control group. And then we have PBS-injected control animals without tumor. And only the cachectic group here loses weight. All the rest remains reasonably stable over the course of the experiment. And we typically have tumor weights that are matched between the groups, and you can see body weight loss, loss of lean mass, loss of fat mass, as well as loss of muscle and adipose tissue masses, specifically in these cachectic animals. This is the C26 model, and then we like to compare this to the SW480 model. The SW480 cells are human colorectal cancer cells, so they are implanted in immunocompromised mice. And once again, they induce roughly 10% body weight loss. And this is reflected at the level of fat mass, but also to a smaller extent at the level of lean mass. Same here with muscle size and adipose tissue sizes, and tumor sizes are reasonably small in this model. So I'm just highlighting here that in both cases, we have a loss of body weight, lean mass, and skeletal muscle mass, as well as adipose tissue mass. So these models are suitable models to study cancer-induced muscle atrophy and cachexia, and they are reasonably similar to the human situation. Now, as Laure mentioned, my background is in adipose tissue biology and in studying adipocyte lipolysis. So I want to spend like 3 slides or so introducing a little bit on adipocyte lipid handling in cachexia. These are rather old stories, but still I think it's worth mentioning. So what we found in a very quick summary was that cachectic adipose tissue was characterized by high lipolysis. And this was also previously seen, for instance, by Martina Schweiger and the Zechner lab in Graz. And we have also seen that, but this wasn't the full story in our hands. But but in addition to seeing increased lipolysis, we also saw an increased re-esterification of fatty acids. So a simultaneous activation of lipid breakdown and lipid buildup. And obviously, this isn't a very energetically favorable situation, so we lose a lot of energy there. And this cycling, this enhanced cycling of lipids in the adipose tissue really contributes to loss of adipose tissue mass in cachexia. And you can see that if we prevent this cycling, we can prevent loss of adipose tissue. This is sufficient to extend survival of these tumor-bearing animals substantially, as can be seen here. And what we're doing currently is that we are following compound development together with industrial partners, because we found that these prototypic compounds that we've published also by now, this is by Honglai Ji in the lab, these prototypic compounds are also effective in human adipocytes, counteracting this lipolysis lipogenesis cycling. So we think this can be potentially, we can move forward with that in cachexia therapy. So these studies showed that preventing adipose tissue wasting counteracts cachexia. I want to spend 1 slide on mentioning that we don't see any browning in cachectic adipose tissue. And I know that goes against many published reviews, but we've never seen it. And let me just show you some rather old data now from our 2016 paper. Browning of adipose tissue occurs, for instance, when animals are under cold stress. Browning leads to increased lipid burning and heat production by adipose tissue. And then a classical marker here is UCP1, and you can see UCP1 staining here very classically in this cold induced adipose tissue. But it looks completely different in the cachectic animals. Yes, we have smaller adipocytes and they might look brownish, but that's actually because we have less lipids and more connective tissue, and we don't actually see any browning. When looking at any kind of marker for browning, there's actually a very significant decrease in these browning markers over time of cachexia development. So we have basically, UCP isn't expressed to begin with to a very high extent in white adipose tissue, but then it's even lower in cachectic adipose tissue, and this is basically true for several other markers of browning. And last not least, when we induce cachexia in UCP knockout mice, there's absolutely no change in comparison to wild-type animals. So UCP is really dispensable for cachexia. And there isn't any evidence of this classical browning cascade, at least in our animals. So what happens instead? This is a project by Honglai Ji in the lab. He's following up on this lipolysis lipogenesis cycling, and one key factor that we identified to play an important role there was AMPK. And Honglai is currently looking for novel AMPK interaction partners in the adipose tissue that can possibly be targeted by this intervention that we have produced as a prototype previously. And he found some very interesting novel roles of AMPK here. And this is something that we are currently following up and hopefully finishing this year. So moving away from the adipose tissue now, we wanted to know if what we see in adipose tissue is kind of, something isolated, or if adipose tissue is perhaps part of a larger kind of tissue coordinated response to cachexia. And in order to understand that, we performed metabolomics from C26 cachectic animals, here in dark red, pre-cachectic animals and tumor-bearing control animals. You can see already from this picture that the metabolites are mostly changed in the cachectic situation and not so much in the tumor-bearing controls. And we did metabolomics of 8 different tissues or 7 different tissues in plasma, and performed a hierarchical clustering because we wanted to know, without any bias, without any, just really in a data-driven approach, what happens in these several different tissues in cachexia. And what was the most interesting perhaps was that all the tissues from cachectic animals clustered in this analysis. And this told us that, the tissues lose they lose their identity. So for the tissues, it doesn't matter so much anymore if they are if they're adipose tissue or if they are muscle or whatever. But it matters more to them that they are cachectic. So this is really what this hierarchical clustering told us. And we really wanted to know then what these commonly altered metabolites were, and what was so special about this shared metabolic tissue across tissues. Shared metabolic signature across tissues, sorry. This was a project that was run together with Andrei Kuda in Prague, who is really an expert in metabolomics and lipidomics, as well as tracing. And we had a more wider look at this data and found that all the tissues that we looked at contributed to all the different clusters. So, metabolite high, very late in cachexia, or metabolite low, very late in cachexia, or some intermediate profiles or so. But it wasn't that, only one tissue contributed the most, or, or the tumor contributed the most. No, it was really that all tissues somehow contributed here. And the biggest changes were really seen in amino acids, in amino acid analogs, and the top 10 metabolites contributing to this cluster here are shown there. And if you have any kind of your favorite metabolite, go to our online tool. It's published in this paper here. It's an online tool. You can just type in your metabolite of choice and then have a look how it behaves across the different tissues in cachexia and in response to cancer. So this was a kind of an unbiased analysis, but we wanted to know even better what this, what was so specific about these tissues from cachectic animals. So we looked at commonly regulated metabolites across different tissues, and then focused on all the metabolites that were altered in the same direction in 2 or more tissues. And this is the full list here. Maybe you can find your favorite metabolite. I don't know. But what I would like to highlight is that a good half of these metabolites here are amino acids and derivatives. So this is in red here. The direction of change is always the same across tissues. Even if it's not significant, then you have, the same direction of regulation. And it doesn't make a difference if we compare cachexia to PBS animals or cachexia to non-cachexia animals. So the tumor isn't so important for these metabolic changes. It's really more the cachexia that is the most important here. And the same is then true for the upregulated metabolites. And once again here, a good half of them are amino acids and analogs, but the downregulated ones here were more like protein-building amino acids like serine, glycine, glutamine, and so on. And here on the upregulated side, we have more modified amino acids, such as dimethylglycine, acetylated amino acids, methylated amino acids, and so on. So that was really quite interesting to us. So What these profiles show is that metabolite regulation is coordinated across tissues because we always have, the same directionality, and it's linked to cachexia, not cancer. So what's going on? We performed a pathway analysis of these commonly regulated metabolites. Here are the top hits: pyrimidine metabolism, folate metabolism, arginine biosynthesis. And then to the right here are the top 10 key metabolites. And it took us a while to see that, but actually, once you see it, you can't unsee it. 8 out of these 10 key metabolites, as well as all the top hits in this pathway analysis, actually belong to the 1-carbon metabolism. So we have here mostly products of 1-carbon metabolism that are coordinately changed, so changed in the same direction across the different tissues in cachexia. So it's really 1-carbon metabolism that is very typical, very classical for cachexia. So what is 1-carbon metabolism? It's, it does a lot of things. It's involved in purine and pyrimidine synthesis. It's important for DNA synthesis. It's important for polyamine synthesis, for methylation reactions, so methylation of amino acids, nucleosides, but also lipids. And it's important for redox balance, as it's also involved in glutathione synthesis. And it's a complex pathway. We have identified 20 metabolites that are either substrates or direct or indirect products of this 1-carbon metabolism. We named them 1 to 20. You can look up your favorite metabolite again later if you want. But then just remember 1 to 20, these are all metabolites linked to 1-carbon metabolism. And if you then zoom in to these metabolites 1 to 20, across the different tissues, and shown here are examples for plasma, liver, and fat, you will see that, almost all of them are really significantly enriched, specifically in the cachectic setting here in dark red, and really not so much in all other groups. And I would like to specifically highlight here products of methylation reactions, such as sarcosine, dimethyl lysine, and dimethyl glycine, that are really consistently, really significantly upregulated across tissues, as well as metabolites belonging to pyrimidine synthesis, such as thymidine here. We then teamed up with Dominic Luther from our center, not from our institute, from our center, to integrate our metabolomics data with matched RNA sequencing data. So these are RNA sequencing of all the different tissues of exactly the same animals that we used for the metabolomics analysis. And what we did here was that we integrated the most significant changes across different tissues, across the different methods. And the top hit here is seleno amino acid metabolism, and that's basically just another word for 1-carbon metabolism. So really, this RNA sequencing confirms the enrichment of 1-carbon metabolism signatures across cachectic tissues. We then used this RNA sequencing information to define or produce a cachexia signature gene set linked to 1-carbon metabolism, and this contains methyl transferases, key enzymes in polyamine metabolism, glutathione metabolism, and the folate cycle. And I'll get back to that in a second. And we then also leveraged this multi-omic analysis to look for an upstream regulator, so something that might be responsible for all of these changes. And the top hit here was LPS, lipopolysaccharide, or inflammation. And this was really interesting because, of course, inflammation is very important for cachexia. And in particular, IL-6 has in the past been shown to be highly important for cachexia, especially in the C26 model, which we have employed for this initial analysis. So we teamed up with Martina Schweiger in Graz, who has previously performed an experiment on knocking out interleukin-6 in C26 cells, as well as with Laure Bindels, who you all very well know, who has treated C26 animals with IL-6 neutralizing antibodies. In order to really understand the contribution of IL-6 or inflammation for these 1-carbon signatures. And I'm showing you data only for the IL-6 knockout today, but it's the same for the antibodies. We can see in this experiment run in a different lab that the same cachexia signature gene set that we have previously found was also enriched here in liver and in muscle, for instance, in dark red, and then it's basically completely reverted by knocking out interleukin-6 from tumor cells. This is true for liver and muscle. And then these metabolites 1 to 20 that I've shown you before are once again really significantly enriched across the different tissues in the cachectic state and then reversed by removing IL-6. So this really shows that inflammation is a key driver in this enhanced 1-carbon metabolism. We paired this analysis with a glucose tracing experiment. So we injected animals with a stable isotope tracer for glucose 1 hour prior to sacrifice in fasted animals. And what you can do by this is that you can follow the glucose all the way through glycolysis and the TCA cycle, or I should say the carbons from this labeled glucose all the way through glycolysis and the TCA cycle. And by the amount of labeled carbons in, for instance, citrate here, you can approximate how much glucose was used for these steps. So for glycolysis or for the TCA cycle. And we did that across all the different tissues once again. And the most striking observation was that we had significantly reduced levels of unlabeled TCA cycle metabolites, shown here by citrate in black, and at the same time a significant enrichment of labeled citrate, and also in particular of high isotopologues. So this means that more carbons were labeled in citrate, and this means that this cycle was actually running faster. So the TCA cycle was running faster because more carbons have been labeled. This was true in all muscles, including the cardiac muscle that we checked. And it told us that cachexia causes a rewiring of glucose flux in these muscles. This is just more data to show you that, it wasn't only citrate and it wasn't only soleus muscle. It was, so these are data for the heart. Once again, you see this very significant enrichment of labeled carbons in these TCA cycle metabolites. You can calculate fluxes from that, or let's say approximate fluxes from that, and calculate enzyme activities. And you can see that glucose flux through glycolysis is increased in these cachectic muscles, as well as TCA cycle activity is increased. We then linked this glucose hypermetabolism, or wanted to link this glucose hypermetabolism to the 1-carbon observation from before. And in order to do that, we went to the cell model. So we treated C2C12 myotubes with increasing doses of methionine. And methionine can overactivate 1-carbon metabolism. And you can see that this is linked to atrophy. So this very well matches our data in mice. And increased levels of methionine reduce the amount of glucose in the medium surrounding the cells. So more glucose is used, and it also reduces the pH. So once again, more glucose is consumed by these cells in which 1-carbon metabolism is overactivated. And this is also linked, or this is also shown by once again applying this glucose tracer, where we have again more labeling of TCA cycle metabolites such as citrate. So 1-carbon metabolism activation is linked to glucose hypermetabolism also in myotubes. And then the other way around, we tried to block 1-carbon metabolism, and we did that by applying FIDAS5. That's a methionine S-adenosyl transferase inhibitor. We can see that this induces hypertrophy in these myotubes and reduced glucose consumption. And perhaps even more importantly, it restored IL-6-induced muscle atrophy. So in dark blue is IL-6-induced atrophy. And then in the lighter blue, you can see that this is rescued with the increasing amount of FIDAS, and the glucose consumption is also rescued. So blocking 1-carbon metabolism causes hypertrophy and reduces glucose consumption and rescues L6-induced atrophy and hypermetabolism. We don't know about in vivo yet. This will have to follow. I think in vivo it will likely be a little bit more complicated, and I'll tell you why. We see this glucose hypermetabolism specifically in muscle, in skeletal and cardiac muscle, and not anywhere else, not in any other tissue. And this is despite the coordinated activation of 1-carbon metabolism across tissues. So the hypermetabolism is specific for muscle, but the 1-carbon signature is not. So that's a coordinated response. And we don't know how these two things go together yet. And it remains to be seen how we can target 1-carbon metabolism and in vivo, and how that will influence glucose consumption in muscles. And another side note that I would like to make here is that the signature gene expression and metabolite profile was confirmed in 6 additional cachexia models. We were asked by the reviewers during the revision process to reproduce our data in 5 additional models, and the reviewers were so kind to name the models which they wanted to see. So this was quite some work, but I mean, luckily it worked out, and you can actually see that the 1-carbon signatures are stronger when there's stronger cachexia, and they are weaker when there's weaker cachexia. So this was actually very nice addition and very nice suggestion by the reviewers. It took us by surprise though. Just some data to end this part of the story here is from the SW480 model that I introduced before. Once again, you see that the SW480 cells induce cachexia, so weight loss and loss of muscle and adipose tissue size. And they induce this cachexia signature gene set in liver and muscle that we had defined based on RNA sequencing from the C26 model. Once again, we have this huge increase in metabolites of the 1-carbon metabolism in plasma, but also across different tissues. And once again, we see glucose hypermetabolism in muscles. So the humanized SW480 model recapitulates all data from the C26 model. And luckily, then we were able to team up with Olga Prokopchuk from the Technical University Munich, who has produced the Munich Cachexia Biobank over the past couple of years. And she has biopsies from liver and skeletal muscle of patients with cancer and with or without sarcopenia. And you can see here that the combined signature gene set is also significantly increased in both liver and skeletal muscle of these patients. So this was Pauline's project, who is here. So I might direct questions to Pauline later, let's see. But basically, in summary, what we found by integrating data from metabolomics, RNA sequencing, and glucose tracing was that there was a coordinated increase in 1-carbon metabolism specific for cachectic tissues. It was driven by inflammation and associated with muscle glucose hypermetabolism. We validated this in cells, and we also validated this across several different mouse models, including a humanized cachexia model, as well as to some extent in patients. This was the end of the first study that I would like to introduce. I still have a couple of minutes to present another one. And the background of this other study that I'm introducing now is that a couple of years back, together with my colleague Mauricio Berriel-Diaz at the Helmholtz Center in my institute, as well as Marilia Seelander in Sao Paulo, we have performed a lipidomic screen of plasma of patients with cachexia. And we found that there was a very specific signature of lipids in the circulation that was high. And these were specifically ceramides and modified ceramides. We compared this human data with mouse data from 4 different cachexia models, and thereby ended up with 6 different ceramides, or with a signature of 6 different ceramides that was always high across different models and as well as patients, specific for cachexia and not, as you can see here, in non-cachectic cancer. There were also 2 LPCs that were significantly downregulated across the different models and patients. So we showed here that ceramides are biomarkers of cachexia and cancer, but are they more than biomarkers? We didn't know that then. And this was exactly the idea behind this project here. And I was very happy to collaborate with Bert Blau in Padova on this advanced cachexia experiment, because here we were able to reach a body weight loss of more than 20%, which is not allowed to do on our animal licenses in Munich. But it was possible to by teaming up with Bert, and we needed that because we wanted to treat animals with a ceramide synthesis inhibitor, and we needed a little bit of time to do that. And our experiments are typically over very fast, so we needed this extra bit of time that we could get by keeping the animals longer with a higher weight loss. What we found was that inhibiting ceramide synthesis by myriocin significantly increased final body weight in comparison to the initial body weight. It did not change tumor size, and it significantly increased liver weight. And the myriocin was able to reduce ceramide levels in liver, plasma, and muscles. And specifically here, the ceramide species that I showed you before, the 16-0 and the 24-0, were elevating cachexia and then significantly reduced by myriocin treatment. Now, what do they do to the rest of the tissues, to the rest of the body? Ceramide synthesis inhibition does not change adipose tissue loss, but it significantly reduced heart cardiac atrophy and muscle atrophies, so measured by several different muscle types. And perhaps more importantly, it made this reduction in grip strength induced by cachexia much less severe. On the molecular basis, the myriocin treatment reversed this induction of atrogenes that is typically induced in the cachectic state in the C26 model across the different muscle types as well. And it also reduced ubiquitination, indicating that there is less protein breakdown. So the global inhibition of ceramide synthesis improves muscle wasting in cachexia. But what is the origin of these elevated circulating ceramides in cachexia? We approached that by measuring gene expression of various different enzymes that are important for various bits of this ceramide synthesis pathway. And once again, here teaming up with Olga and the Munich Cachexia Biobank, we performed a qPCR analysis of liver, visceral adipose tissue, subcutaneous adipose tissue, and muscle of patients with various degree of weight loss. So we have mild cachexia, 2% weight loss, 2 to 5% weight loss in sarcopenia, or more than 5% or more than 10% weight loss. And you can see from these graphs here that the significant enrichment of ceramide synthesis enzymes was only observable in the liver and not in adipose tissue and muscle. And there was a significant correlation between body weight loss and the expression of these genes, specifically in liver and not elsewhere. So liver expresses high levels of ceramide synthesis enzymes in patients with cachexia. And indeed, we confirmed that also ceramide levels were high in these livers, in these human livers, and also the species that we found before. And there was a significant correlation with body weight loss and the amount of ceramides in the liver, as well as a correlation between the amount of ceramides and the expression of these ceramide synthesis enzymes. And the same is true for modified ceramide. Mechanistically, we wanted to find out what these ceramides do. They've previously been involved in in many different processes, including inflammatory processes, but also mitochondrial dysfunction. So we wanted to find out what ceramides do for cachexia. And by performing unbiased a proteomic screen in liver of cachectic animals, we found that the most affected pathways here were mitochondrial pathways. So cachexia-induced reductions in mitochondrial translation, oxidative phosphorylation, electron respiratory function, and so on, increased in mitochondrial dysfunction. And you can see the myriocin effect is to reverse all of this. So it's really rather black and white that ceramide synthesis inhibition restores mitochondrial function in liver. And we can see this also at the level of mitochondrial morphology. So these are TOM20 stainings for mitochondria. You can see that in the C26 animals, there is reduced branches, reduced junctions, shorter branch length in the cachectic state. And this is completely restored by the addition of, or by the treatment with myriocin. And by electron microscopy, we see a very similar thing. We also see a very similar thing by Western blotting of OXPHOS proteins, as well as measuring enzyme activity. So this is pretty solid evidence that ceramides actually impact mitochondrial function in liver. And this global ceramide synthesis inhibition improves cachexia by improving liver mitochondrial function. So what happens if we liver specifically target ceramide synthesis? We did that by treating animals with a virus targeting SPT2, so one of the key enzymes of ceramide synthesis. And this virus is specific for hepatocytes. So by only blocking ceramide synthesis in hepatocytes in a cachexia experiment, we can prevent to a certain degree body weight loss, and we can improve grip strength, and we can improve mitochondrial morphology in the liver. And perhaps even more importantly, we can improve mitochondrial function in muscle. And that was a bit of a surprise. We found that in collaboration with Anja Zeigerer in Heidelberg and Carlo Viscomi in Padova, who did the mitochondrial enzyme activity assays. But it was quite obvious that the liver-specific inhibition of ceramide synthesis was able to restore mitochondrial function in muscle. This was data for myriocin, but this is data down here for the liver-specific targeting. And then lastly, also in vitro in the cell culture, we can show that ceramides induce mitochondrial dysfunction and atrophy in myotubes. So here, increasing doses of ceramides lead to increasing concentrations of cleaved caspase 3, increasingly impaired mitochondrial respiration and atrophy in a dose-dependent manner. So this is the summary of the second study, is also led by Pauline. We show that tumor-secreted or pro-inflammatory factors increase circulating ceramides, and this is associated with impaired mitochondrial function in liver. And we can block this either by global inhibition of ceramide synthesis or by liver-specific blockage, and then this improves cachexia, and this is also the case in patients. So in summary, there are coordinated host responses as well as tissue crosstalk factors exemplified by ceramides that contribute to cachexia, and these are potentially targetable mechanisms that we want to pursue in the future. And with this, I would like to thank my whole group. The people who have contributed most to the studies that I showed today are highlighted in bold, but obviously we have a lot more projects ongoing. I would like to thank my funding agencies, and most importantly of all, you for your attention. I'm hoping to still have time for questions. Thank you.
Thank you very much, Maria, for this very dynamic, exciting talk. We have a lot of time for questions, so thank you for sticking up with the time. So the floor is open for question. You can either raise your hands and then you'll get to ask your question live, or you can put your question in the chat. There's already one question in the chat that I'll ask to Dr. Rohm. So this question comes from Dr. Franciella Morena. She's saying, amazing talk and study. You showed that unlabeled TCA intermediates are markedly lower in cachectic tissue, but this represents 80% of the pool in the other groups. So could this suggest that control muscles relies more on alternative carbon entry into the TCA, such as anaplerosis, rather than indicating only reduced TCA flux? Since amino acids like glutamines can contribute to anaplerosis and are depleted in cachectic tissue, could this reduction restrict those alternative inputs?
I mean, ideally, in order to find out about TCA flux, you would want to do a glutamine tracing, right? But yeah, we haven't done that yet. It's planned for the future. In principle, I would like to highlight that we did a 1 hour tracing, and that's a very short time. And we are following up now with longer tracing, and I think then we will also see a higher labeling in general. I think, I think what happens is that because the total level of these TCA metabolites is very low in cachectic muscles. So what happens is that they really have a, dampened metabolism. And then the second you inject glucose, they metabolize the glucose like crazy.
So it's a very interesting point. I was wondering, did you include some per-fed animals, to control for this effect of glucose depletion? Because we know those animals, the cachectic one, they have hypoglycemia.
So in this study, we used only fasted animals. So all animals were fasted for 6 hours. But I should also say that we have not systematically included a pair-fed control. So we have not controlled for kind of long-term fasting or long-term effects of reduced food intake, which is typical for this model. Yes.
Yeah.
But there is a study on bioRxiv from the lab of Tony Hui, who did that and who looked at that. Yes.
Perfect. I see that Francesca is raising her hand, so I'll give her the floor. So let me check how I can do that. Franciella, you should be allowed to speak now if you can unmute yourself if you want.
Oh, no, I was just going to say thank you for your answer. And you said that you guys have plans on doing glutamine tracing as well. Is that correct?
Yeah.
Okay, thank you. I'll look forward to knowing how that goes.
No, I mean, we are definitely following up on this in this direction as well. I think it's important to understand better what exactly is going on. And this was, a first attempt at doing that, but this is definitely not the end. I think there's much more to learn. And the tracing is such a powerful methodology that, I think we can learn a lot from that in the future.
Thank you again.
So I'll take a question from the chat. We have Devat Kandoria, my apology for the pronunciation, that is congratulating you. Phenomenal work. According to your experience, would one carbon metabolism be affecting anorexia in cachexia?
Anorexia. I don't know. Would it? I mean, we haven't done that experiment. What you would want to do would be to in vivo target several different key enzymes of the different steps of one carbon metabolism, see how that affects body weight loss in general, but also aspects such as food intake or glucose hypermetabolism and so on. But yes, we haven't done that yet, so we don't know.
And then we have Selma Masri that is raising her hand, so I'll give her the floor. Selma. No, Selma, you should be allowed to speak. Yes.
Yes, awesome. Can you hear me, Maria?
Yes, I can hear you. Hi.
Hi, beautiful work. Yes, beautiful work. Absolutely fantastic. I have a couple questions. So the first is, I'm very struck and intrigued by the connection between the one carbon metabolic cycle and the hypermetabolic phenotype you're seeing in terms of glycolysis. Given the role of one carbon metabolism in so many pathways, did you guys also look at some of these additional pathways and what's going on, if there's additional links? And then also, there are amino acids that feed into one carbon metabolism. Have you looked at some of the sources of this, if this could be coming, for example, from muscle breakdown that could be helping fuel one carbon metabolism?
Mm, the later question first. I'm fairly sure that this is what's happening. Yes, that we have a breakdown of proteins and thereby freeing of amino acids that can enter this cycle. And we don't see any serine labeling from glucose at this short time, but it would be really interesting to extend the time of labeling and then potentially see serine labeling. What I can tell you, that's like data that we got, I think 3 days ago, is that when we induce atrophy with different means, so independent of cachexia, then we can, and then do a 24 hours tracing with glucose, then we actually see increased labeling of serine associated with atrophy. So in a completely different cachexia independent model of myotube atrophy, we also see that increased one carbon metabolism. So I thought that was really interesting.
Yeah, that's super interesting. Beautiful work, Maria.
Thank you.
So we have a question from Mariana de Carvalho. Thank you for your amazing presentation and what an amazing work. I was wondering about all metabolites that are breakdown from muscle and adipose tissue and that are transported in the blood. Does the liver absorb them, or the inflammation is somehow affecting its function? And is there an hepatic steatosis somehow? Thank you.
We don't have typically at end stage, we don't have hepatic steatosis, but I'm not excluding that we could possibly have that at earlier time points. I think at the time point when we look, these mice are just depleted of lipids. So they don't have lipids in adipose tissue and they don't have lipids in liver either. But I would assume that hepatic steatosis is a transient thing. I mean, it's also, we've seen that a couple of years back, or published this a couple of years back, that you can see hepatic steatosis in some conditions in cachexia. So, but I think it's a transient thing. And the same is true for myosteatosis, actually, that Sander Rensen's group has recently published. And then, yeah, I mean, unfortunately, we don't know about the fluxes yet. So what goes into the muscle, what leaves the muscle. I think you need different technologies, different methods for that. We'll follow up on that in the future. We have some funding for that, actually.
Good. Looking forward to it. So we have two questions. We have one question from Subharajan Das that is stating, nice presentation. What do you think if we inject glucose deprivation resistant cells in xenograft mouse model? Is there a possibility of cachexia?
Glucose deprivation. Sorry, can you say that again?
Yeah, I'm trying to reformulate it. I think the idea would be to administer to mice cells that would be resistant to glucose deprivation, and so asking if this would be inducing cachexia. That's my understanding.
It's a very interesting suggestion. I don't think this has been done, but it would be very cool, of course, right, to follow on something like this, to really systematically see what happens if I alter one by one key key metabolic pathways in the tumor cells, and then how does that influence cachexia development? I think it's a very cool suggestion. Yes, thank you, but I don't know the answer.
And then the last question for today, I'm checking the time, coming from Zaven Kabakian. My apology for the pronunciation. Great talk. At the precachectic stage, are there any metabolic change that can be used as a predictive biomarker for early therapeutic intervention?
I mean, that would be the gold standard, right? To have that, to be able to do that, and to be able to, in patients, diagnose cachexia earlier, and then be able to open up to earlier intervention. I think what we saw from these ceramide studies was that they were actually increased early on. So they were actually increased already in the precachectic state. Metabolites, it wasn't so clear. So that right now, I would say that this ceramide signature was the best prognosis, but that was in mice. I wouldn't be able to tell in patients. We're not there yet. I think we would need big cohorts, where the cachexia is really well characterized over time, and then kind of take blood samples over time and then see how disease trajectories develop. So this would all be kind of future ideal situations, but we are not there yet. But in mice, the ceramide signature is really already high in the precachectic state and can predict disease severity later on.
Perfect. Thank you so much. So with that, I would like to thank you again on behalf of the Education Committee from the Cancer Cachexia Society for your time and for your all the science that you share. Thank you very much for everyone who attended, and we are looking forward to host you again in a follow-up seminar. So thank you very much, everyone. Enjoy your day.
Thank you so much.
Thank
Bye.
you. Bye.